Single-cell datasets from multiple batches often have different cell type compositions. Batch A captured mostly T cells. Batch B captured mostly macrophages, a method that aligns all cells across batches would force T cells and macrophages to overlap, destroying biology. Integration methods must align the same cell types across batches while keeping different cell types apart. Harmony operates in pica space. It doesn't touch the gene expression. Start with all cells in a shared pico embedding. Cluster the cells. For each cluster, compute the centroid per batch. Push each batch's cells toward the shared cluster centroid by a small correction factor. Re-embed and repeat. After a few iterations, cells of the same type from different batches overlap, while different cell types remain separated. Harmony is fast, it scales to millions of cells and effective for most integration tasks. It's become the default first-pass integration method. Mutual nearest neighbors takes a different approach. For two batches, find pairs of cells where cell A from batch 1 is in cell B's nearest neighbors in batch 2 and cell B is in cell A's nearest neighbors in batch 1. These mutual nearest neighbor pairs are the bilingual speakers, cells of the same type that appear in both batches. The difference between paired cells defines the batch correction vector. Hide this vector to all cells in the batch, weighted by distance to the anchor pairs. Scanorama extends MNN to multiple batches by finding overlapping cells across a panorama of datasets. He handles batch through the generative model, the hidden orchestra's scVI architecture. The encoder maps gene expression to a latent space Z. Batch identity is fed to the decoder as a one-hot covariate, but not to the encoder. The encoder never sees which batch a cell came from. It must encode only biological variation into Z. The decoder uses both Z and batch to reconstruct the gene expression, learning how batch affects the counts. The latent space C is batch corrected by construction. scIB adds cell type labels for semi-supervised integration, using known annotations from one batch to guide the correction. How do you evaluate integration quality? SHAPE, the single-cell integration benchmarking framework, measures two competing objectives. Batch mixing. How well are batches mixed after correction? Measured by scIB and batch AS. Biology conservation. Are real biological differences preserved? Measured by cell type AS, enemy, and isolated label score. A method that perfectly mixes batches but merges distinct cell types as failed. A method that preserves biology but leaves batch artifacts has also failed. The best method score high on both. Practical guidance. Foo batches with balanced design, same conditions in each batch. Use combat or include batch as a regression covariate. The simplest solution. Many batches with similar cell type compositions harmony. Fast, effective, operates in pica space without modifying expression. Complex integration across technologies, for example, 10x and SmartSeq from different studies scVI. The most flexible, handles different count distributions, learns nonlinear batch effects. Always verify, after correction, do known cell type markers still distinguish the expected clusters? If not, you've overcorrected. Pre-labs, 3 dialects, combat recalibrates means and variances, works for bulk and balanced designs. Harmony iteratively aligns clusters in pica space, the workhorse for single-cell RNA-seq. MNN finds bilingual cells to define correction vectors. scIB learns a batch-free latent space through the scVI generative model. The translator removes the technical dialect while preserving the biological message.